Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. We already talked about a lot of organelles related with the cell biology. Now, this is another video lecture which is also related with the cell biology. And in this video lecture, we are going to talk about plasmid. Okay. So, first of all, what is plasmid? Plasmid is actually an extra chromosomal material that are present in a prokaryotic cell, not in a eukaryotic cell. So, let's suppose if this is a bacterial cell. Inside a bacterial cell, what we have a genetic material. This is representing the genetic material of the bacteria, and with this genetic material we have what? We have an extra chromosomal material which are usually in a circular form. Now this extra chromosomal material are called what? Plasmid. Okay. Plasmid is a very important genome for the bacteria surviving. When the bacteria have plasmid, so they will be survive in their community, in their environment, in their population for a longer period of time. Because they produce a special kinds of genes, a special kinds of protein, we can say means because these means they have, because we can say that actually this plasmid have a special kinds of genes. Such genes are important to produce a special kinds of the proteins. Now once they produce a special kinds of the protein, then such protein is used for a bacterial survival. Okay, so this plasmid is usually, we can say, it is a double-stranded DNA. Okay, as you are watching, but it will be in a circular form. Okay, now every bacteria have a single plasmid, but in some bacteria, for example, B. burgdorferi, such bacteria have only 9 circular DNA and 12 linear DNA. Okay, so in this case, we have a bacteria, we call that B. burgdorferi. So let me write the name. B. burgdorferi. Okay, so in this case they have 12 linear and 9 circular. Okay, so 12 linear and 9 circular plasmid. Now let's talk about the types of the plasmid. So if we see the types of the plasmid, so we have different types of the plasmid. So let's talk about the type of plasmid. So the first types of the plasmid, if we talked about, that's about the circular plasmid. Now circular plasmid are those extra chromosomal material that are usually present in a prokaryotic cell, which are in a circular form. For example, this is a circular chromosomal material, or we can say this is a circular DNA. Now this is a circular plasmid as well. Such circular plasmid DNA are self-replicating, okay, and produce a special kinds of genes which are specially means important for the bacterial surviving. This is about the first. The second, we have the linear plasmid. Now linear plasmid are those plasmid which are in a linear form. For example, let me draw here. For example, it would be in a linear form. Okay, means this is what? This is a linear genome, or we can say this is a linear plasmid. In linear plasmid, there are special kinds of sequences, or we can say special kinds of genes at their end, or we can say at one of their end. Such genes or such sequences are used to provide protection, or we can say such genes or sequences are important to prevent the plasmid from the degradation and permit it to replicate. Okay, now this linear plasmid have less than 30 genes. Okay, so they have less than 30 genes. Then we have another plasmid, we call that let me write episomes. Now, episomes is another types of the plasmid which are integrated with the bacterial chromosomes. So let's suppose this is a bacterial chromosome. So in this case, the episomes will be present at somewhere of the bacterial chromosomes. Okay, it is also a plasmid, but such plasmid will be integrated with the bacterial genomes. Okay, so when the bacterial genomes are divided, or we can say when the bacterial genomes are replicated, after their replication, this chromosomes or this plasmid will be also replicated, or we can say with the replication of the bacterial genomes, the same plasmid will be also replicated. Why? Because it is integrated with the bacterial genome. Okay, so this is called what? This is called episomes. Then third we have curing. Third we have curing plasmid. Fourth we have curing plasmid. Now, curing plasmid is also in a circular form, and it is those plasmid which can transfer their same characteristics to the other daughter plasmid. Okay, so if let's suppose this is a plasmid which is in a circular form. Okay, now this plasmid have a mutagenic plasmid, or we can say a mutation phenomena is occur in this plasmid. So the same characteristics will be transferred to their daughter one as well. Okay, so usually plasmid can produce a single copy of the another plasmid. So we can say that curing plasmid are those plasmid which can transfer their characteristics to the daughter one. Okay, so if this plasmid is mutagenic, so then the same characteristic will be transferred to the daughter. If this is healthy plasmid, so the same characteristic will be transferred to their daughter. Okay, or if there is some problems or mutation occur, so such plasmid we can treat it with the help of UV radiation, ionizing radiation, antibiotics, and optimum temperature, and acridine mutagens. Okay, with the help of these materials we can treat such plasmid as well. Now, once we treat it or changes, or once we bring some changes, or once we treat this plasmid, so they can also be replicated without any changes or without any treatment. Okay, so whenever we want to treat or whenever we want to change, so they will be replicated with these changes or with this treatment, but without this treatment and changes they will also replicate. Okay, then after that we have another plasmid, and that's about the conjugative plasmid. Conjugative plasmid. Now, conjugative plasmid, as the name suggests, it is that plasmid which have a special kinds of genes that produce a special kind of hair-like structure. For example, they can produce a hair-like structure. So let's suppose these are the hair-like structure. Okay, so these hair-like structures we can also call that what? Pili. So such such plasmid have a special kinds of genes that produce a pili, and this pili is important to produce a kind of conjugation tube. Okay, or we can say it is that plasmid that can produce a conjugated tube, because they have a special kind of genomes that are responsible for the production of conjugated tube. Now, this conjugative plasmid can transfer their copies to another bacteria. Okay. After that, we have another plasmid. We call that F plasmid or F factor, or we also call that fertile plasmid. So let me write the name. F plasmid, F factor, or fertile plasmid. Fertile plasmid. Now, as the name suggests, this is that type of the plasmid which are actually used for the fertilization. Now, this plasmid is usually present in E. coli, and they have a special kinds of genes that produce sex pili. Okay, so it is usually present in what? It is usually present in E. coli, and produce what? They can produce sex pili. Sex pili means these are the pili, and once they produce the 6 pili, so due to this 6 pili, the other bacteria will be attracted. For example, let's suppose this is a bacterial cell, and let's suppose this is F positive bacteria. Now this F positive bacteria have F plasmid, or we can say they have a fertile plasmid. Now this fertile plasmid will produce a kind of sex pili. So this is a sex pili. Due to this 6 pili, at the other hand, we have another bacterium. Let's suppose this is another bacteria. This is F negative. Now due to this 6 pili, this bacteria will be easily attached to this to this one. To this F negative. Okay, so we can say this 6 pili are actually used for the attachment to another bacterium. Okay, so once they attach to F negative, let's suppose this is F negative bacterium. So once they attach to the F negative bacterium, so then they will transfer their characteristics to the other bacterial cell. Now this we can say this F plasmid can produce 1 to 3 copies a plasmid. Okay, and the size of this plasmid will be 95 to 100 kilo base pair. Okay, so this is about their size. Next we have another plasmid. That's about R plasmid, or we can say R factor, or we can also call that resistance plasmid. As the name suggests, R plasmid or resistance plasmid, so it is that kind of the plasmid. It is also in a circular form. It is that kind of the plasmid that are specially used to produce a kinds of genes that are responsible to destroy or to change the antibiotic structure. Okay, so if let's suppose this is a plasmid, so they will produce a special kinds of genes. So let's suppose these are the genes which are produced by resistance plasmid. Let's suppose this is R plasmid. So these genes will destroy the antibiotic. If this is the antibiotic, so they will destroy the antibiotic. Okay, they will sometimes destroy the antibiotic, or they will modify the antibiotic. Once the bacteria are destroyed or modified, so then this antibiotic will be not working. Once the antibiotic are modified or destroyed, so then such antibiotic will be not working inside a bacterial cell. Okay, we can say that this plasmid is very important. So when a bacteria have such plasmid, so bacteria will survive for a longer period of time, because this plasmid will give protection to the bacterial cell. Okay, not only that, this plasmid will also we can say it is dangerous for the human cell as well, for the human body as well, because when bacteria have such plasmid, so it is very dangerous bacteria for us, because we cannot treat this bacteria with the antibiotic as well. Okay, then we can use very high dose of antibiotic, then such such bacteria will also kill, but it is not possible. Okay, now resistance plasmid have actually single genes, but in some cases, or we can say in some bacteria, they have more than 8 genes. Okay, moreover, this plasmid is not integrated with the bacterial chromosome. As we talked about in episomes, the plasmid will be integrated, but in this case, this resistance plasmid will be not integrated. Okay, so this is very important. Now this plasmid is usually present in Pseudomonas, Shigella, and we can say Proteus and some other gram-negative bacteria as well. The size of this plasmid will be, if we talked about the size, so the size of such plasmid will be 54 kilo base pair, and they also produce 1 to 3 copies of their plasmid. Okay, bacteriocin encoding plasmid, Now bacteriocin encoding plasmid are those plasmids which can produce bacteriocin proteins. Okay, so they can produce bacteriocin proteins, and such bacteriocin proteins are actually responsible to kill the other bacteria or to kill the other cells. We can also say that So we can say if the bacteria is present in a population, or if the bacteria is present in a microbial population, let's suppose, and such bacteria have this kinds of genes, so then they such bacteria have this kinds of plasmids, so then this bacteria will produce bacteriocin proteins, because they have special kind of genes that code for the bacteriocin proteins. Then bacteriocin proteins will produce, let's suppose this is a bacterial cell. So what actually happen? A bacteriocin proteins will produce a kinds of this bacteriocin plasmid produce a kinds of proteins. We call that bacteriocin. Bacteriocins are actually make a pores in a bacterial cells. Another bacterial cells. Okay, once they make a pores another bacterial cells, so then such bacterial cell will be what? Such bacterial cell will be die. Moreover, they can also weak the peptidoglycan layer of the bacterial cells. Okay, if you don't know about the peptidoglycan, so I already explained it. Okay, so just go back to my channel and watch it. So they can actually weak the peptidoglycan layer. Moreover, this bacteriocin not only kill the peptido, means they not only kill the bacterial cell, we can also say they can degrade the DNA and RNA. Okay, so let's suppose this is the DNA materials, and so we can also say that they can also attack on the DNA, means the DNA are also degraded by this bacteriocin as well. Okay, so it is very important. It is also important for the bacterial survival, means this bacteriocin encoding plasmid are also important for the bacterial survival. Okay, then we have another plasmid. Let me erase this one portion. So after bacteriocin encoding plasmid, then we have another plasmid. We call that cold plasmid. So this is number 9, cold plasmid. Now this cold plasmid are that plasmid which have a special kinds of genes that can code for colicin. Okay, so colicin is actually a protein that are actually means it is a protein that are specially against for E. coli. Okay, so we can say they can produce a colicin protein. So cold plasmid are also, means this cold plasmid are also sometimes we can say it is also a conjugate plasmid. Why? Because in this case they will also produce a 6 pili, but except 6 pili they will produce also a colicin protein as well. So that's why we can say that this cold plasmid is actually a conjugative plasmid as well. Moreover, this cold plasmid have also carry the resistance genes which are show the characteristics of this resistance plasmid as well. Okay, so this cold plasmid, if we see, so the size and the size of this cold plasmid will be 25 kilo base pair. Now such plasmid are usually present in a Enterococcus faecials. Okay, so let me write Enterococcus faecials. Okay, so try to best remove the spelling mistake by yourself. Then we have another plasmid, we call it virulence plasmid. As the name suggests, we also call it pathogenic plasmid as well. Now virulence plasmid are that plasmid which can make the bacteria more pathogenic. Why? Because they have a special kinds of genes that produce a proteins we call that enterotoxin proteins. Okay, such enterotoxin proteins can make the bacteria more pathogenic. Okay, so this plasmid is actually used to make the bacteria more pathogenic. Now the size of this plasmid will be 83 kilo base pair, and they are usually present in enterotoxogenic strain of E. coli. Okay, last we have metabolic plasmid. So let me write metabolic plasmid. Now metabolic plasmid are those plasmid which are specially used to degrade the sugar and some other large molecule, for example sugar, carbohydrates, proteins. Okay, so such plasmid are used to degrade a large compounds. Okay, so such plasmid are actually used to degrade a large aromatic compounds like proteins, sugars, carbohydrates. Okay, so they can degrade like that materials. Now if we see, so the size of this plasmid will be 230 kilo base pair. Okay, and it is usually present in Shigella, Pseudomonas, Klebsiella, E. coli, and Salmonella. So in this bacteria, these metabolic plasmid are present. So that's all about the plasmids. Okay, so I hope you understand about the plasmid and the structure of the plasmid and everything about the plasmid. But if still you have any kind of questions related with the plasmid, so write it into a comment. Inshallah, I will try my best to give you answer as soon as possible. Okay, so if you like this video, so please make sure to hit the like button, share this video and subscribe the channel. Okay, and also press the bell icon to get more videos lecture regarding with the cells. Thank you so much. Take care. Allah Hafiz.